Hi, I'm Professor Nick, and today I'm going to teach my student Sam about protein synthesis. And right now we're going to start with transcription. Okay. Inside of a cell there's a nucleus, and inside that nucleus an RNA will associate itself with a base sequence of one strand of a gene. Other enzymes will unwind that gene and then will leave a portion of the gene exposed, which then the RNA will move along the exposed gene and polymerize an mRNA molecule. The nucleotides will be the exact same as the gene copy, or the codons will be the exact same from the gene that the RNA is copying it from. When the RNA reaches the end of the gene, the newly found mRNA molecule will release itself into the nucleus and then escapes through a pore in the nuclear envelopment, then will enter the cytoplasm. Now I'm going to draw you a picture to help explain this further. This is the nucleus of the cell, and there are many genes in the cell that look something like this. Then once an enzyme RNA attaches itself to one of these genes, and the enzyme will expose a piece of the gene, which will then be an opening in it, like so. And the codons will be out like so, and then the RNA will come down, attach itself to the codons, copy those codons from that gene. Then once it is done, it will release itself from that gene and be free in the nucleus, and then will exit through a pore from the nucleus into the cytoplasm. Catching everything so far?
Yeah, so it just leaves the pore then, the RNA?
Yes, leaves because RNA itself, no, mRNA will exit through the pore, because the RNA itself can't exit through the can't exit the nucleus. That's why it transferred into mRNA, and that's why it leaves the nucleus. Alright, now we're going to start talking about transcription. Okay, first off, now tRNA will come into play, come into play. Its job is to transfer amino acids to the ribosome, where they are bounded together by the enzymes in the exact sequence specified by the gene and by the mRNA. There are 45 common types of tRNA, each being capable of carrying one of the 20 or more common types of amino acids to the protein synthesis site. Now, in this diagram here in the textbook, you can see there's like amino acids, and the tRNA will attach itself to the acid like so.
Okay.
Then wait to be enveloped by the ribosome, which I will get to talk to you about right now. Okay. When the first tRNA has maneuvered itself into the correct position at the beginning of the mRNA message, then the ribosome moves the mRNA strand along, bringing the next codon into position to be read by another tRNA. As the amino acids are brought to their proper position along the length of the mRNA, they are joined together by enzymes. As an amino acid is bonded to the chain, its tRNA is released and moves away from the ribosome to pick up another amino acid and will be ready to be absorbed again. Okay, so you see as the amino acids and tRNA go in, they're attached to the mRNA strand by enzymes in the ribosome.
Okay.
And then when they are released, the tRNA is released out of the ribosome completely, like you can see it leaving there, and it will go and find another amino acid to attach itself to and get ready to do that stage again next time it is needed.
Okay, I see.
And then after all this, the last stage is when the mRNA strand is finished, it will snap off. Then everything that is in there, the tRNA, the amino acid, and the ribosome, will all disband from each other, and then they will be ready to be used next time when they have to do this again. This happens so fast, it's probably like 20 that just happened there as many times as I snap my fingers once.
Really?
Yeah, that's how fast it's always happening, and so fast. Everything, yeah, and that is basically protein synthesis. Do you have any real questions? Or any questions at all?
Not too much. Just to get so there's in total 4 different NAs then, or?
There's many types of
there's an mRNA, a tRNA, right? The RNA. So are those the 3 we've gone over then?
No, we also went over DNA inside of the nucleus. That's where the main copy of the coding came from. But those are the 4 main types of RNA. There was RNA, DNA, mRNA, and tRNA are the main ones that I talked about today.
And then the mRNA, that was like the messenger one, right?
Yep.
That's
actually the name of it. That's what the M's for, messenger RNA.
Okay, I see.
That's the actual name of mRNA. Alright, anything else?
No,
Alright, and there's a picture I drew him. If you can see it, it's not too good, but I'm not an artist. Then I also used this diagram as a textbook, which were very helpful. And that is everything. Maybe next time we'll learn about cell structure or something. I bet.